तो उसके बाद तो
हेलो
लैपटॉप चेक होगा ना।
हेलो, आवाज आ रहा?
नहीं आएगा, अब आएगा, अब जाएगा।
हेलो, आ रहा है?
यहाँ नहीं आएगा, वहाँ
ठीक
जाएगा।
है।
यहाँ
अच्छा।
पर
चैट
कर ली।
अच्छा।
Hello.
Hi, sir. Hi.
Hello. Good to
Okay.
see you.
Yes, sir. So good to see you. Thank you so much for your time. How are you doing?
Very well. Very well. Crazy times. How's the situation in India?
So crazy.
Yeah.
Same here. So he's Professor Vijay. He's our Dean Academics.
A
And
pleasure to
yeah.
meet you.
Welcome, Dr. Frank Tucker. It was really our privilege to have you on the very first webinar series that we
Oh,
are
really? It's
hosting.
the first one. Oh my God.
Okay, before we start, let me welcome, good afternoon to all the viewers. And let me tell you briefly about our institute, the Institute of Liver and Biliary Sciences in New Delhi. So ILBS, we call it in short, is a autonomous super specialty medical cum research institute for the treatment of liver and biliary diseases under the government of Delhi. It was established in 2010, and it has been really, we have been trying hard to attend benchmark in areas of both patient care as well as research. So these are the excuse me. The interruption. So this year, because ILBS was established in 2010, so this year we are celebrating 10 years of its existence and holding such activities in such unprecedented corona times. That's something important. So we planned, I think the initiative was taken by my colleague, Dr. Savneet Kaur. And this webinar series is called as From Basic Liver Biology to Clinical Implications. So for this first lecture, as I told earlier, we have Dr. Frank Take, who is currently the chairman of the Department of Hepatology and Gastroenterology at Charité University Hospital in Berlin, in Germany. We are extremely grateful to him for sparing his precious time for this webinar. Dr. Take is an MD PhD doctor who received education and training at the medical school in Hanover in Germany and at Baylor College of Medicine in Houston, Texas, USA. Since 2006, Professor Take has been a clinical physician and research laboratory leader at the University Hospital Aachen in Germany. He is board certified in internal medicine, gastroenterology, endocrinology and diabetology, critical care medicine, and infectious diseases. He has served on the governing board of the European Association for the Study of the Liver, that is EASL, since 2014 and has been instrumental in formulating 2017 EASL guidelines on the management of hepatitis B virus infection. Dr. Take's research interests are clinical and experimental hepatology, particularly monocyte macrophage biology and liver diseases. He has done pioneering work on the mechanisms of inflammatory responses in liver diseases, dissecting immune cell subsets, driving progression to fibrosis, targeting these mechanisms, and translation into clinics. He has been involved in several clinical trials, and he has published over 350 peer-reviewed original and review articles and received several prestigious awards, including Emmy Noether Funding Award from German Research Foundation. Now I invite Frank Take to deliver his talk on new insight into liver macrophages in NASH pathogenesis. Professor Take.
Thank you very much for this kind introduction. Let me see if we can make the... Can you see the presentation? Is that working for you?
Yes, we can see.
Wonderful.
Yes.
Okay, and you can hear me all right?
Yes.
So thank you very much.
Absolutely.
It's really a great privilege to be the first one on this lecture series from the really prestigious ILBS Institute, which is well known worldwide, particularly, of course, for your research on liver failure and the clinical input you provided to the community. I would like to give you today and discuss with you some new insights into liver macrophages and NASH pathogenesis. And I have some disclosures which partly relate to the fact that some of the pharmaceutical companies support our research with providing us with compounds and sometimes also with funding to translate our findings on macrophages and liver disease. What I would like to discuss with you today is a little bit about NASH, meaning non-alcoholic steatohepatitis, and what the role of inflammation and fibrosis is for the progression of the disease, the particular role of macrophages and myeloid cells as important immune cells in the liver, and the therapeutic implications of these myeloid cells, but also on maybe some drugs for the regulation of inflammation and fibrosis. Why do I think this is important? If when I was serving on the EASL governing board, the European Association for the Study of the Liver, Tom Carlson and myself looked a little bit at our congresses, and over the years, we noticed that there are some areas which had major breakthroughs, like the hepatitis C virology, which with the new DAA compounds that are now declining, and of course the rate of progress that is being made. But one field is really booming in the past years, and this is NAFLD, non-alcoholic fatty liver disease. And why is that? It is because the demand is so high. We're now facing the consequences of an unhealthy lifestyle, particularly in the Western countries, and maybe it will become even more prevalent also in Asian countries, in India, with obesity, metabolic diseases, dyslipidemia, insulin resistance, which also affect the liver. In Europe, it's estimated that about 25% of the general population have non-alcoholic fatty liver disease, which of course is not always like a very aggressive disease, but about 10% of this fraction have non-alcoholic steatohepatitis. And these are patients that are at risk of developing liver cirrhosis or hepatocellular carcinoma. And in particular, the proportion of patients with NASH, the steatohepatitis, among patients with fatty liver disease is increasing. And so there are projections for the next 15 years, for instance, in the US, that the number of patients affected with NASH cirrhosis will more than double within this foreseeable period of time. And the same is also true for Germany, although we believe we have a healthier lifestyle than the Americans, but we're still very much affected by non-alcoholic fatty liver disease. And what is the population that is at greatest risk of progressing with the disease? We have many studies showing us that the stage of liver fibrosis is actually very crucial in this regard. So there are, for instance, here data from a meta-analysis of 5 studies with paired biopsies, patients that are being followed up for a long period of time. And the stage of fibrosis was closely related to all-cause mortality, but also to liver-related complications and mortality. So this explains why there is a big hype among the pharmaceutical companies to develop new drugs against non-alcoholic fatty liver disease. And a major target of these drugs is to prevent progression of fibrosis or even induce regression of liver fibrosis. And you can see here a list of compounds that is not even complete in different stages of clinical trials. And of course, the hepatologists among you are well aware of recent results from phase 3 clinical trials that showed in an interim analysis after 72 weeks of treatment, promising anti-fibrotic effects for obeticholic acid, farnesoid X receptor agonist, that showed, but also quite discouraging negative results for selonsertib. The trial was stopped. Gilead-based ASK1 inhibitor failed to prove efficacy in NASH, and elafibranor, a PPAR alpha delta agonist, also was recently reported to fail its primary endpoint. So it's really important that we understand the mechanisms, the underlying pathophysiology of non-alcoholic fatty liver disease. And actually, I think we can learn a lot about it for many types of liver diseases. If you think about it in a very simplistic way, there is, of course, a connection if you have an altered metabolism. So you have fatty liver that can cause inflammation, and inflammation will then drive fibrosis, which means in the liver, the activation of hepatic stellate cells and myofibroblasts to produce extracellular matrix. But the picture is a bit more complex. We also understand that inflammation changes the metabolism. So inflammatory signals can also alter the way lipids are handled and glucose is handled in the liver. And we also understand that a lot of the metabolic pathways are directly involved in fibrosis, which is partially also true for the FXR pathway that is a target of obeticholic acid. And if you think about how metabolism, inflammation, and fibrosis are connected and integrated with each other, then the macrophages play a crucial role, because this is a cell that can receive signals from the hepatocytes, from the outer environment, and also from other inflammatory cells, and can then promote inflammatory mechanisms, but also fibrosis. And we know for quite some time now that macrophages in the liver are a key characteristic of virtually any type of liver disease. For instance, if you treat mice with carbon tetrachloride for 6 weeks, you induce liver fibrosis, which you can see here in red with collagen fibers. And along these fibers, you see an accumulation of macrophages, here stained by F480 as a macrophage marker. When you look closer with modern technology, for instance, you do a FACS analysis, you appreciate that the cells that look like macrophages actually consist of different types of population or different cell populations. So you have a heterogeneity among macrophages. And one of the key findings was that macrophages actually have two sources. One is the local resident macrophages, which we nowadays call Kupfer cells, and the other is monocytes that can be recruited upon inflammatory signals that have a different source or cellular origin. And that will also impact a little bit on their function. However, we understand now that the differentiation of macrophages, so their functional profile, whether they would promote inflammation, promote fibrosis, or would downregulate or dampen inflammation, is very much dependent on environmental signals that they receive from the direct environment, but also from systemic signals. And there is no really M1 or M2 type macrophage, but it's rather an integration of many, many signals. And this is work that very convincingly show that the macrophages adapt their phenotype to the signals they receive. Another aspect that I would like to highlight is that the macrophage phenotype is not necessarily stable, meaning if you change the microenvironment, a macrophage that would promote inflammation and would promote fibrosis can also become a repair macrophage that would promote regeneration and resolution of disease. And we are starting to understand the signals that would switch functions of macrophages at the moment, which might be very, very helpful if you think about new therapeutics. So to just maybe bring this together, the macrophages are located on the sinusoidal side of the liver, the Kupfer cells of the liver sinusoids. They can be augmented by monocyte-derived macrophages, here abbreviated MOMF, that can be recruited to the liver upon signals. And the macrophages are thereby optimally located to receive different signals and integrate signals, both from the liver as well as from the gut-liver axis, as well as from the circulation. And one example how we can understand the function of the macrophages is if you have liver injury. And for instance, one of the most prevalent acute hits to the liver that would lead to acute liver failure is acetaminophen or paracetamol that would affect the hepatocytes, that they release danger signals, which we call DAMs, danger-associated molecular patterns. And one of the immediate responses of the Kupffer cells is that they release CCL2, which is a chemokine that would then attract new monocyte-derived macrophages to be recruited to the liver. And we have studied this, for instance, in vivo, and these are data derived from an intravital to photon microscopy setup, where we have the mice anesthetized, then surgically expose the left liver lobe, then place the mice under the 2-photon microscope. And we also give them IV fluids and keep them ventilated to have a stable anesthesia. And that allows us to understand the interaction of different macrophage compartments. You can see here on the left side in a control mouse in green monocytes that patrol through the liver. These are CCL2 expressing cells that can respond to chemokines like CCL2. And in red, you see the Kupffer cells that are relatively stable in the liver. And upon acute liver injury, you see that the two populations of macrophages are still distinct. You see Kupffer cells in red, and you see monocyte-derived macrophages in green, but you see a dense accumulation of the monocyte-derived cells. So they are able to augment the pool of macrophages in the liver and immediately form clusters that could then respond. If we sort from these mice with acute liver failure, the different populations of monocyte-derived macrophages and Kupffer cells, you can appreciate that the Ly6C high MOMF, which is the population of freshly recruited monocyte-derived macrophages, are actually the ones that express a lot of pattern recognition receptors that are very responsive to changes in the environment. They're not so much different in the expression level of cytokines or chemokines, but really very much in the way they can respond to signals they receive from the environment. And this is something we are exploring further. And one technology that we do quite a lot, and where we might really have collaborations with the ILBS Institute, is that we do a multiplex immunostaining, which allows us to visualize those interactions between different cell populations. You can see here staining of control mouse, liver fibrosis mouse, or a NASH model, which is a choline deficient amino acid supplemented high-fat diet, that we see in the different colors, different cell population. We can stain more than 10 different markers at the same time that help us to understand the polarization of different macrophages and also the interaction with or between different cell types. This is another example that we can use the same technology also for human liver to understand really the organization in the tissue and the interactions with the cells. Of course, it's also important to then understand the function of the different cells. And if we go back to a mouse model of NASH, meaning the methionine choline deficient diet, and sort the different populations of macrophages, mainly the Kupffer cells, the resident macrophages, and the monocyte-derived macrophages, you can appreciate that they nicely separate by RNA seq analysis from each other. And we see in the Kupffer cells a lot of genes related to innate immunity and lipid metabolism. So we believe that they are very much orchestrators of the disease by recruiting new immune cells and by also participating in metabolic operations in the liver, whereas the monocyte-derived macrophages are more related to fibrosis angiogenesis. So they very much influence the stroma or the non-parenchymal reaction of the liver in NASH. Of course, technology advances, and we have now the ability to do single-cell RNA sequencing to understand the heterogeneity of hepatic macrophages or myeloid cells in general. And the first experiments we did was to isolate from the liver and the bone marrow in parallel in a high-fat diet model with little fibrosis, but a lot of NASH and steatohepatitis, different populations of leukocytes where we enriched for macrophages, meaning we excluded the excluded the lymphocytes, excluded the neutrophils, and then did a single-cell RNA sequencing using the 10x chromium system. What we can appreciate then is that we not have only two populations of macrophages, the Kupffer cells and the monocyte-derived macrophages, but that we actually see a lot of diversity within the macrophage pool, which I think helps us to understand why the cells can have so many different functions. We see at least 3 populations of monocyte-derived macrophages. We see a remarkable difference in the dendritic cell populations, and we also see Kupffer cells. And if we compare healthy mice or healthy liver Joe diet fat mice with Western diet fat mice with a high cholesterol, high fructose diet, you can see that particularly the populations of monocyte-derived macrophages are very, very much enriched in the disease, in the diseased liver. And if we look at the different macrophage populations that we see, for instance, the monocyte-derived macrophages, we are able to identify certain marker sets for the different populations. And we're also able to understand their polarization. And one thing that was very apparent is that among all monocyte-derived macrophage clusters, we see a certain downregulation, for instance, of S100, A8, A9, which is upregulated in the normal diet, downregulated in the Western diet. And this is a phenomenon that affects almost all myeloid cell populations. So you see here a list of the different populations that were identified in Chow and Western diet. And there is a very, very similar regulation of gene profiles among different populations of myeloid cells. So we believe that high-fat diet or Western lifestyle induces a very common myeloid phenotype, meaning a particular aspect of inflammatory myeloid cell polarization, not even only in the liver, but also in the bone marrow. If we find that in mice, we can isolate those cells, and this phenotype seems to be relatively stable. We can keep the phenotype in the culture and can transfer those polarized cells also to other mice, which then determine the functional responses to the injury. But of course, this is not only a phenomenon in mice. It's easier to study in mice than in human, but we can also now look at several data sets from human liver disease. For instance, here data set where we participated in human liver cirrhosis. And if we apply single-cell RNA sequencing technologies to human liver, we can appreciate also a large heterogeneity among the non-parenchymal cells. And one particular aspect was that in the macrophage population, we do see a particular subtype of scar-associated macrophages that is also TREM2 positive. And this can also be visualized by immunohistochemistry and immune fluorescence. At this fibrotic fibers, we do see this macrophage subtype. However, what is difficult to understand from these data, and that's the limitation, they are very much descriptive. And in order to understand the function of the different populations of cells that we identify, it's then important, to my understanding, to go back into mice. And for instance, for the TREM2 positive macrophages, there are some good indications from other groups showing that they actually might be beneficial. So scar associated does not mean that they promote fibrosis. They might also have a beneficial function. For instance, they reduce excessive inflammation after acute liver injury. And they also, in an obesity model, TREM2 associated adipose tissue macrophages counteracted inflammation and metabolic disease in these obesity models. Of course, you may ask, so how does all the knowledge, the understanding of this particular cell type of the immune system help with the management of the patient, especially with patients with advanced disease, with liver fibrosis or even cirrhosis? And there are some ideas. If the monocyte-derived macrophages that are recruited to the livers are a key driver of fibrosis, you could think about medication to reduce the recruitment of these cells to the liver. Another aspect is, of course, if you identify regenerative macrophages, you could isolate them or maybe induce this phenotype in vitro with autologous macrophages and then reinfuse them into patients. And you could also say, if I understand what drives the beneficial polarization of macrophages, you could maybe pharmaceutically induce a repolarization of macrophages towards repair and regeneration. And all these aspects are currently being explored in clinical trials. Maybe to the first aspect, what I showed you from the mouse model was that the monocyte-derived macrophages that are recruited to the liver are actually the ones driving fibrosis. So they release factors that activate hepatic stellate cells that then release extracellular matrix. And it's very tempting to speculate if you can block this, block the recruitment, that you would then also block fibrogenesis. And there is a way to do that. There's a drug called sinicriviroc. It's an orally available dual inhibitor of the chemokine receptors CCR2 and CCR5. This drug had been initially developed to treat HIV due to the CCR2 inhibitory function and had been tested before in quite a number of patients with HIV. If we look in NASH, fibrosis and cirrhosis, we actually do see that with the progression of from steatosis to NASH fibrosis and cirrhosis, there is an increase in CCR2 positive macrophages in the liver. And you can see here in brownish, the CCR2 expressing monocyte-derived macrophages in the liver. So the rationale is that you block the recruitment of these monocyte-derived macrophages in mice. That can be easily done by feeding the mice on a methionine choline deficient diet, then treat for the last 4 weeks with sinicriviroc. And we do see a tremendous reduction of the monocyte-derived macrophages in the liver. That would be the red circle in the FACS plots. And this actually translates into an interesting phenotype. So what we do not affect so much is the extent of steatosis or steatohepatitis. So this is apparently also driven by the Kupffer cells, but we tremendously reduce liver fibrosis in those mouse models of NASH and steatohepatitis. So there was a clear rationale from the mouse experiments that if we block the recruitment of macrophages, we reduce liver fibrosis. And we have tested this also in a phase 2 clinical trial, which is now completed, where we randomized patients to either placebo or CBC, then did a liver biopsy after 1 year. And then after a second year, there was still randomization of placebo to CBC after year 1 to have a higher number of patients benefiting from sinicriviroc. The data are very much reflective of the mouse experiments. So we do not affect steatohepatitis or transaminases or body weight with blocking the monocyte-derived macrophages. But there was a higher proportion of patients improving by fibrosis by 1 stage after 1 year, and after 2 years, at least a trend of patients improving by more than one stage, meaning 2 stages or more. And this is being tested at the moment in a phase 3 clinical trial. Maybe towards the end of this year, we're going to see first interim results after 1 year of treatment, whether this can be reproduced in a large cohort of patients. In total, there will be about 2,000 patients being treated with this drug. The other aspect that I told you, can we use maybe macrophage infusions as a cell therapy? There's a nice work from Stuart Forbes' group showing that this in principle possible. He treated a small number of patients, published very highly in Nature Medicine, basically showing that this is feasible. There was not really a clear therapeutic effect of macrophage therapy. It was more proof of concept study where the adverse events were recorded. So it was a safe procedure. You can actually isolate macrophages from patients, culture them towards a restorative phenotype and reinfuse them into patients. It's not so clear if that is really beneficial or not. It's also not so clear to me whether the macrophages remain at this reparative phenotype once you infuse them, or if they just die and then exert indirect effects in the liver. The other aspect for therapy would be to try to repolarize the macrophages that are already there in the liver towards a restorative phenotype. And this is an interesting concept because the macrophages could, in general, be very beneficial also in hepatocyte metabolism and could induce also apoptosis of stellate cells or revert the stellate cells to a quiescent phenotype. And this is the matter of ongoing research, which you might call immunometabolism, that you think about how to influence the macrophages so that they are beneficial on metabolism, but also macrophages, if they become activated, they also change their own metabolism. So for instance, an activated macrophage that is inflammatory would activate NF-kappa B, glycolysis, ROS production, certain transcription factors, form inflammasome, produce cytokines. And a lot of the drugs that are being tested as metabolic drugs in the field of NASH, for instance, agonists of PPAR pathways, drugs that are meant to influence fatty acid sensing, for instance, inhibitors of GPR84 or inflammasome inhibitors, would not only affect hepatocytes, but would also have a tremendous effect on macrophages. And we have explored this with lanifibranor, which is a pan-PPAR agonist. So PPARs are transcription factors that have different isoforms, alpha, gamma, and delta, and that are quite cell type specific. And what we saw is that if we treat mice on a CDAA high-fat diet model with lanifibranor versus selective PPAR agonist, we can actually integrate the beneficial effects of the different isoforms. And it had not only an effect on metabolism and steatosis. You can see the H&E stains here with the vehicle, the large lipid droplets, with lanifibranor, almost no lipid droplets, but it also had an effect on macrophages. And so in this work that had just been accepted by Journal of Hepatology, we were able to show that the different PPAR isoforms actually affect different cell populations. And in the hepatocytes, at least in the mouse system, PPAR alpha was most relevant. And the macrophages, PPAR delta is the one that drives a beneficial activation, and PPAR gamma was the one to downregulate stellate cell activation. So the idea is, of course, if you have selective PPAR agonists, you could maybe impact different cell populations. But if you have the pan-PPAR agonist, you could also have beneficial effects on inflammation and fibrosis, which you might not have with a very, very selective PPAR agonist. And with this, I think there are many open avenues elucidating further the role of macrophages in the pathogenesis of NASH. I'm very grateful to my team in Berlin that helps us to explore this. You can see here like a very initial picture when we moved to Berlin with a small group. We're constantly expanding. Very happy to collaborate also with colleagues from India. We have now Mohsin Hassan, who's Indian, recruited from a lab in Bern. We have Cornelius Engelmann, who was with Rajiv Jalan before on ACLF. And hopefully we can establish a close connection between the labs in New Delhi and in Berlin. With this, I would like to conclude. New insights into liver macrophages in NASH pathogenesis. I think it's important to study this disease and understand metabolic liver diseases because they will be the leading cause of cirrhosis, transplantation, and cancer in the near future. And fibrosis is actually the mechanism that is considered very much decisive for the prognosis. Macrophages are very, very important because they're key cells integrating different signals, and they impact inflammation, injury, stellate cell activation, fibrosis, angiogenesis, but they are also important to resolve inflammation and fibrosis, both in mouse models as well as in man. We do have now new technologies like single-cell RNA sequencing. We have imaging techniques like intravital microscopy that allow us to dissect which population of cells is important at which stage, what is the exact mechanism activated in a beneficial or in an aggressive macrophage. And this can be translated into new therapies. I showed you the example of the chemokine receptor antagonist, cenicriviroc. There are other ideas how to repolarize macrophages. I think a lot of the NASH drugs like elafibranor, lanifibranor has also implications on macrophage biology. And of course, maybe even macrophage transfer as proposed by Stuart Forbes will have a place in the future for the treatment of patients with advanced liver diseases. And with this, I would like to thank you very much for your attention. Thank you for joining, and I'm happy to answer your questions.
Thank you so much, sir. Thank you. Thank you for this wonderful talk and so many open avenues, as you rightly pointed out. So we can have some questions.
Yeah, I think what you are proposing in New Delhi is very important, that we use the functional data that we have from mouse models to translate that what is relevant and active in human disease. And this is exciting, and you have a great opportunity to study human liver disease. And I think that is the natural.
Yes, sir.
Hello, Chief.
So,
Right. Sorry, I joined late. Please continue with the discussion. Just wanted to say hello.
Thank you very much. It's really an honor to send my greetings to New Delhi.
Savneet, please continue.
yes, sir. Yes, sir. So, so do we have questions? Okay.
Let me ask a question that what may be the reasons for failure of autologous transplantation of macrophages?
Yeah, I'm personally very skeptical. First of all, if you use in a mouse model just monocytes from the bone marrow, which are relatively immature, and you give them in a mouse model of fibrosis, it would aggravate the disease. So if you have very immature cells that then come to an injured liver, they would rather promote inflammation, aggravate the disease. If you have a very like terminally differentiated macrophage, that would be very beneficial and promote resolution of disease. I think those cells are probably yeah, they won't stay alive. They may reach the liver or get stuck in the liver, but they would just die. And maybe that is a beneficial signal. There is, I think, in the first issue of Nature this year, there was a cardiac fibrosis model where they also did cell therapy in a mouse model and showed that it was a bystander inflammatory effect that was beneficial, but not a beneficial effect from the macrophages per se. I think they don't reach the liver alive.
There was
Just another quick, yeah.
sorry, there was one paper by Stuart Forbes in
Yeah.
Nature Medicine. 6 patients or so, they had given the macrophage therapy and a huge
कैसे दिखे?
brand. I was wanting to have your opinion on what do you think and about the work, and is that the way forward for us?
Yeah, I think I was actually a reviewer of that Nature Medicine paper, and I have to say I was very skeptical because they do not show efficacy. What is really, I think, amazing that they showed they can do it. So like having this logistics behind to be able to isolate the cells, infuse them. They infused only one time. That is probably not enough for the patients. At least it shows that this is a concept that could be done and could be further developed, but I think they failed to show efficacy. So I'm not convinced that the cells were alive, that they did anything good, but they did not do any harm. And to establish the proof of concept that one can do a clinical trial is, of course, valuable.
Yeah. I think another day we can have a long discussion on these therapies, but today I think Savneet and Dr. Vijay may ask more questions of the research group, please.
Yes, sir. So, Dr. Take, there's a question by a viewer that will the allogenic monocyte transplantation be functional in a cirrhotic microenvironment? So I think...
Yeah, that also raises the question if the macrophage is so adaptive and responses to the microenvironment, will you be able to kind of fix it in a good phenotype, transfer it, and then it still does something good? And I'm not sure that will happen. I think it might be possible if you're very terminally differentiated as a macrophage, but then the likelihood that the cell will die is very high. If you keep the cell in a yeah, let's say immature state, they will just adapt the phenotype. So I think the environment will then determine what the macrophage does and not the opposite. And what you want is, of course, that the macrophage tells the environment, okay, resolve fibrosis. And I'm highly skeptical. It works in mice, one has to say, but even there, the cells do not stay in that phenotype very long. Yeah. And of course, mouse models are much more dynamic. So I'm not convinced it will work in patients. And it's definitely no solution for a large group of patients because it's extremely laborious to do that.
Yeah, and so many mouse models, what do you think is the best? Because like now there are reports saying that MCD models are not good for studying metabolic changes. And I mean, for NASH, probably we hardly get cirrhosis in NASH models. So do you think, I mean, in this area, we really need some more of experimental studies?
Yeah, I think the difficulty is that with the mouse models, they all have limitations, and I think you need to understand the model quite well where to use it for. So if you want to understand the mechanism of fibrosis in NASH, I think the MCD model has some value because you have a very stressed hepatocyte that then induces inflammation and fibrosis. And with a Western diet model, you can wait for a very, very long time, but you get only very mild fibrosis. So it's not a good model to study mechanisms of fibrosis in NASH. And so I think there is no perfect model. And people tend to switch now to the CDAA high-fat diet model because it's a good compromise. You do see steatosis, steatohepatitis, and fibrosis, and you do not see cachexia as in the NASH models. However, you do not see insulin resistance, as you would see in a traditional high-fat diet or Western diet model.
Okay, so another question is, what is macrophage plasticity difference in steatosis, NASH, and fibrosis microenvironment? I mean, how they switch states in these
I think
pathologies.
we do understand a little bit what is associated with this. For instance, NF-kappa B, inflammasome activation, glycolysis, ROS production, that is all associated with a switch towards inflammatory phenotype. What are the signals that would downregulate that are not so clear. PPR delta is one that would kind of like switch the macrophage back to a more healthy phenotype or repair phenotype. There are some good indications that phagocytosis as a signal would dampen the inflammatory response of the macrophages, which would explain if you inject cells and they die and they are eaten up by macrophages, that this would down-modulate an inflammatory activation.
Okay. So another question is, what is the role of CD36 receptor in macrophage-mediated HSC deactivation?
I'm not sure I can answer that. We have never worked personally on CD36. I'm aware of some good work that has been done in that area. I'm not sure if this is a crucial pathway that cannot be bypassed by other signals like TGF-beta or PDGF or so on, signals that are well understood for macrophages. It's also clear that macrophages and stellate cells do not necessarily physically need to interact. If they do, they can activate stellate cells, possibly also deactivate, but if they are separated, for instance, in a transwell experimental setting, also the soluble mediators of an inflammatory fibrogenic macrophage can activate a stellate cell. Yeah, I'm sorry that I'm not sure about the role of CD36. It's definitely worth exploring.
Okay.
Hello, good evening, sir. So it was a very nice talk, and I have one little question from the same study you saw about the Stuart Forbes study. So, which they are claiming that the macrophage can reverse the fibrosis and improve liver regeneration. And particularly in the cirrhotic model, they have used the allogenic source of monocytes. Sorry, autologous.
Autologous, yeah.
And yeah, and my question is that whether cirrhotic patient monocytes will be as functional as the healthy after differentiation, because they are showing that there is no difference in the healthy and the cirrhotic monocyte after getting the resolving macrophages. But we have some data which we are suggesting that they are not as good as.
Yeah, I think there are some older studies showing that if you isolate monocytes from the blood of a cirrhotic patients and differentiate them to macrophages and then do an LPS stimulation, that they in a large proportion of patients have some sort of immune paralysis, which would explain why patients with ACLF, for instance, or cirrhosis are more susceptible to infections, which then would promote ACLF. And we do see also changes in the macrophage or in the monocyte populations in the circulating blood. For instance, we have many more non-classical monocytes in the blood of cirrhotic patients. So I would fully agree with you that that the monocytes in a cirrhotic patients per se are not healthy. And they are the source, of course, to develop those macrophages. However, if I understand the Stuart Forbes study correctly, they have mobilized kind of stem cells and then did a leukapheresis and then did an ex vivo differentiation of the cells. So relatively complex protocol, and of course they had more cells than just the ones that would circulate normally in a cirrhotic patient. So I would assume that they have more immature stem cell-like cells mobilized from the bone marrow.
But they haven't used the GCSF for the mobilization. They are just using that normal leukophoresis, and they are just isolating the monocyte from them.
Okay.
Because
okay.
one caveat I found in that the patient selection they have taken is a very early stage of cirrhosis. So they haven't taken I think male less than 10 they have taken there.
Yeah, but I think the reason for that is, of course, a safety issue that you need to prove first in relatively healthy patients that you're not. I think the thing they had in mind was like a cytokine storm or so, which you don't want to induce in a patient. And I think that was an obligation from the regulatory authorities that they needed to start with relatively healthy cirrhotics, stable cirrhotics. Yeah. No, but I agree with you. That is not the ideal population to demonstrate efficacy.
Yeah, because I think if you might have read his paper on the ALF in the animal model, recently they have said. So in that model, rather than using as a autologous, they have used the allogenic source. And then they commented there that when they use the autologous mice cells into the mice, that time they found that the response was not good in ALF model. And when they put the human monocyte there, so that means they shows that they are acceptable, they are tolerable in the mice. And when they put the human, and then they show that it is giving a better recovery. Because the first model they have says that this is not possible, but in the second model they are saying, they themselves are showing that this can be possible. So what is your comment on this?
That would fit to the observation that we had in the NASH models in mice, that the phenotype is actually quite imprinted on the macrophages, at least in the liver, but also like in the bone marrow, where of course fatty acids and everything is also present in an obese patient or in an obese mouse. So maybe like healthy volunteers are the better source of macrophages for cell therapy.
So you agree with that this allogenic monocyte transplant is feasible?
I mean, it is feasible if you reach really a high purity. If you have contamination with lymphocytes, I think it's very
dangerous. Then it will be a problem.
It's risky. Yeah.
Okay, thank you, sir.
Anything else? Okay, so okay, one more. So one comment. Anything did you see on endothelial and macrophage interactions? Like a little bias toward endothelial, so.
No, absolutely. The endothelial cells are probably extremely important also for the macrophage development. We understand now if you deplete Kupffer cells, there's a beautiful paper from Martin Guillaume's group in Immunity, I think from last year, showing that if you deplete Kupffer cells, monocytes can engraft the mouse liver and can become regular Kupffer cells or Kupffer cell-like macrophages. And the important signal, like showing the monocyte that there's a niche and a need, is probably coming from the endothelial cells. We don't understand all the signals yet. It's definitely more than just integrins or so that would just attach the cell. So there must be more to it. Yeah, if you work on endothelial cells, we do not have so much experience with endothelial cells. It's definitely a very important aspect to understand macrophages, because part of the signal comes from endothelial cells.
Okay, so another question from a vascular biologist. So we have Jordi with us. So he's asking from Barcelona. So firstly, nice talk. May also macrophages in ACLD, which may have a very different phenotype and actively communication with vascular cells, be partly responsible for the failure of clinical trials in NASH? Should we pay more attention to HMO and other sinusoidal cells?
Yeah, that's That's a very interesting and intriguing question. And probably I'm well aware that I think if you look at our paper in Journal of Hepatology on the PPAR agonist, one cell population that we did not tackle is the endothelial cells. And we found the different effects in hepatocytes and stellate cells and in macrophages. And I think Dr. Gracia Sancho has some nice data that PPAR pathways are also important in endothelial cells.
Yeah.
And maybe part of the efficacy of the drugs, but also failure of the drugs can be related to that fact. We did not study that in detail, but I think there is some important work still going on and to be published soon.
Great. So I think one last question coming.
Two.
Okay, two. Okay, so another question is, a reply from Jordi saying, yes, we must further add the study of L6. Okay, and one last question is the role of macrophage senescence in CLD versus ALF, in acute liver failure versus chronic liver disease.
I'm not sure that has ever been studied really in depth. I'm not aware of the data. Of course, senescence is a very important mechanism in ALF and ACLF, but I guess it has been mostly described for hepatocytes. To which extent macrophage senescence plays a role or not, I think it needs to be determined. Senescence by itself and the senescence-associated phenotype of a hepatocyte, which would, for instance, include the release of CCL2 that then promotes monocytes to be recruited to the liver, is something that we start to understand. To which extent macrophage senescence is involved, I'm not sure, but definitely macrophages respond to senescent hepatocytes, and that is probably a major mechanism in ACLF at least.
Okay, so I think no more questions, and really thanks to you for taking out time in spite of your busy schedules. Really grateful, and I also thank all the viewers, who have joined us.
Soon in person. Let's hope.
Yeah, definitely. I just
evolve collaborative programs.
Yes. I just wish that these Corona times are over and we can have you soon here, and and the project gets funded.
Thank you so much.
Okay.
Thank you so much.
Thank you.
Okay. Bye.
Have a great day. Yeah. Bye.
Two more questions.